So we start from the beginning. Well, even if people are still, more people are now connecting, let's start today first webinars for the Implanta AU Cost Network project. It's really a pleasure for me as coordinator of the project to open this very interesting series of webinars. It's the only solutions that we have now. Everybody is aware of the situations that we are facing worldwide for the COVID infection. And so even if we have budget for organizing meetings in presence, I have the possibility, as we have done in the past period, to have this meeting and conference in presence that were really helpful and useful to get in touch, know new people, and develop new ideas on this new technology. Unfortunately, now it's not possible. And so we found this solution of organizing webinars. And from now to the end of the project. Unfortunately, the project will end by beginning of April 21. So this is the reason why we already think that it will be really impossible to have other meeting to be able to organize other meeting in presence. So until then, we will work with this system, so through webinars. What we have today is the first series of 3 webinars that will be in December before Christmas, so next Monday and the Monday after, focusing on 3 important aspects related to RNA technology, RNA-based technology, actually more near to the pesticide. Next, we are now already working to the program of other 6 and plus the final meeting, conference, always through webinars. So as soon as dates, programs, and speaker will be defined, we will announce the next what will happen in January, February, and March with the new webinars. And I want to say that, however, this is finally an interesting system because as we see from the number already for this first webinar that was announced officially only a few days ago, we got already more than 200 people registered, and now the connected people are already more than 120, and it's increasing. So let's take the positive think of the COVID diseases that beside all the negative things, we have developed these new tools that in some way probably will also help to spread, to have more larger participation to the activities that we have for the eye plant. So I want really to thank everybody, particularly the chairs, the working group chairs, and what we call the core group of the project that help us to set up this system, this program. And I look forward really to listen what will be the presentation of today, of the next, and what will be the program of the next period. And so I wish you a good virtual event. And Jeremy will give you some more detail on how the webinars will be organized. Thank you, everybody, for your participation and for your contribution.
Okay, thanks, Bruno. Thanks, Bruno. So first of all, I'd like to thank all the speakers this morning who are coming from, ranging from Queensland and Australia to United States. So this is why we've had to arrange it at this time to try and make it reasonably convenient for everybody. So you've seen the program, I've just taken it down. We will start with Nina's talk, Nina Mitter from Queensland. And then we will have Kat Capel from Syngenta, Ethan Barnes from Greenlight, and Salvatore Arpaia from RNAi. If you have questions for the speakers, please put them in the chat box. And what I will do is I will have a look at the chat box and I will try and draw together some of the major questions that people are asking. I think all the speakers are quite happy to receive emails, so if at the end of the webinar you still have some unanswered questions and so on, please contact the speakers, or you can send emails to Bruno and I, and we can forward them on to the speakers. So that will be the procedure today. For Nina and Kat, we will have a question and answer session immediately after their talks. I think for the talks by Ethan and Salvatore, we will wait till the discussion at the end, and then we can deal with these issues. Unfortunately, Kat has to leave us this afternoon sometime, and also we don't want to keep Nina from her beauty sleep, so we will let her answer her questions at the end of her talk. So remember, put them in the chat box, and I will have a look at them, and then we'll proceed from there. So first of all, we will have Nina giving her talk. She's talking about the formulations of double-stranded RNA, using bioclays, particularly focusing on viruses. But she tells us that she's been working on fungi and insects as well. So if you have any questions on those applications, I'm sure she'd be quite happy to take them as well. So over to you, Nina, if you'd like to introduce yourself and then put up your presentation. So thanks, Nina. We look forward to hearing you.
Oh, thank you very much. Thanks, Bruno, Jeremy, and Guy for this wonderful opportunity. Yes, all the way from land down under, and that's what maybe COVID can help us do. It's a constant Zooming kind of things, but very grateful for this opportunity and such a beautiful forum. When I was looking at the agenda and the talks, it's really, I think I'll have to give up my beauty sleep and listen to these talks. It's really great, very great session. So thanks once again for making me a part of it. And also, you know, I have a lot of pressure on my head now. I'm the opening batsman. You know, India and cricket and Australia and cricket go hand in hand. So I'm sure, I hope as an opening batsman, I do justice to this innings of the iPlanta webinars. So I'll start a little bit about myself. I am located at the University of Queensland in Brisbane in Australia. And at the University of Queensland, we have an institute within the university, which is called Queensland Alliance for Food and Innovation. And that institute is actually totally focused on applied agricultural applications with a vision and mission for sustainable agriculture for tropics and subtropics. And I'm the director, Center for Horticultural Science at this institute. And my own area of research, of course, is on RNAi-based biopesticides, but I've been working on RNA interference for the last 15, 18 years. And now, since 2011-12, transitioning into this topical application of RNAi. So I'll start my talk. Just give me 1 second to share my screen and praying that everything works well. Can you still hear me? Thank you. And just let me know if in case there's something wrong with any slides, or if you can't see anything or you can't hear me. Hope it all goes well. As Jeremy mentioned, it's about, going to be nearly 11 p.m. here at my end, but let's see how we go. Today I'm sort of going to talk about, of course, the very title of this iPlanta webinar series is RNAi-based biopesticides. And what I'm going to focus a little bit towards is the delivery mechanism, which encompasses delivering the trigger molecule of RNAi using bioclay. I don't think so to this august audience, I need to go into the introduction that why pesticides are important, but they have issues as well, such as issue of development of resistance to pests and pathogens. We also, of course, are aware of the issue of residue, especially working in the space of horticulture. I am really aware of the residue on fresh food and fresh produce, and we are all striving towards that clean, green, safe produce. Not only that, the future consumer has become really aware. In all my interactions now with the consumers, this comes up very clearly, that the consumer of tomorrow actually wants to know the provenance. They want to know how their food is being grown, where it is being grown, and how it is being protected from pests and pathogens. So this biological control or other methods of alternative control assume a greater significance. Another issue that we do, of course, once again are aware of lack of specificity sometimes of these broad spectrum pesticides. And we all know that we don't want to do anything to our beautiful butterflies and moths and monarch butterflies. So we really do want to save our beneficials. And it's not easy to develop new green chemistries. You know, if we are looking for new chemicals, it really is a difficult task. Maybe takes up to about 13 years to register a new chemical crop protection product, and only 1 in 139,000 makes it to the market. So it's hard to find safe green chemistries, and that's where I think RNA interference comes in. When I started working on RNAi, of course, it was for developing disease-resistant plants, especially targeting viruses of vegetable crops. And it did go well, you know, like it's nothing against genetic modification. I'm all for it. You get resistant plants, which you can develop and deliver for the life of the plant. But the question here was, can we deliver RNA as a topical application instead of that genetic modifying the plant? And once again, this boils down to perceptions and acceptance issues for genetically modified plants. But for me, it was more also the cost and time involved, especially when you think of horticultural space and crop protection. It's easy to develop a genetically modified tomato or Arabidopsis or other plants which are amenable to transformation. But if you think of horticultural crops or tree crops or large woody crops, it is very difficult even to have, you know, genetic modification for disease resistance. I personally have gone through this. I also work on avocados in my another area of research, so happy to talk about avocados sometime. But with avocados, when we were trying to make our rootstock resistance to a fungus called Phytophthora cinnamomai by genetic transformation, took me 4 years. I think I tried 800 independent transformation events, and I ended up with 4 plants which were GM, and 2 of them turned out to be chimeras. So it's not that easy also a pathway towards genetic modification, and there is a lack of transformation protocols in this space, and that's what made me more attractive towards the topical application of RNAi or RNAi as a biopesticide. I started thinking of this idea way back in about 2011. And whenever you think of such ideas, you feel, oh, wow, you are the first one, you know, to think about what a clever idea it is. Let's spray double-stranded RNA on a plant and let's see if it works against the viruses. And that was my first call. And then when you do that, very soon you realize, and Google is your friend, and you start looking at the literature, and it becomes very much aware that people have worked on it. And the very first paper I read was by Francis Tenlado et al. From Spain, who published their work in 2001 and 2003, and multiple other papers from then onwards. And this picture is sort of became my motivation. Sorry, I'll go back to that picture, this picture in their paper became my motivation, where they showed that working with pepper mild virus, so they sprayed it with double-stranded RNA, and as you can see, they challenged so this was really encouraging to me to see, yes, okay, topical application of RNAi can work. Since then, if you look at the literature, maybe I haven't even covered maybe all the papers. This is just with viruses, I'm saying. I am trying to actually collate a database on the papers published from 2010 to 2020 on RNA-based biopesticides, and I'll be happy to share once I have collated that data. It really works. There are 12 papers just on 10 different viruses and 11 different host plants and even viroids, which people have published showing researchers all across the globe that yes, it works. And in this slide, you can see it working against papaya ring spot virus by one of the groups. So the concern that I had when I started looking at this literature and very early on, that is, as I said, 2011-12, I thought, why this hasn't gone viral, as in the terminology used these days, although it's a scary terminology these days as well with COVID. But what it brought to me was, what was the issue? Why wasn't this technology really taking it off and becoming a big thing? And then you reach the end of the publication and you realize the issues which are involved from a practical perspective, that yes, double-stranded RNA, although more stable than small single-stranded RNAs, is still unstable under harsh environmental conditions. There could be a degradation by enzymes on the leaf surface. UV and sunlight could be issues. It could get easily washed away. And whatever papers I was reading, it was like protection lasts maybe only for 5 to 7 days after spray. So this wasn't a practical alternative if this technology has to come on board to make a real difference on the ground. And this is one of the paper by Gan et al. Way back in 2010 again on sugarcane mosaic virus, where they did the field trial as well on sugarcane mosaic virus plants in maize. And what they also showed was that, you know, the protection window, if you spray it on day 0, you challenge it with the virus on day 5, you're getting the protection. But if you challenge it with the virus 7 days or 9 days after the spray, you lose that window of protection. So the whole philosophy then was, can we deliver RNAi as a spray, but as a stable application, at least which can last for about 4 to 8 weeks or 12 weeks, providing that window of protection. So the rationale that we started with was, how can we convert naked dsRNA experiment into a commercially viable system for growers? Can we make it stable? The whole application should be degradable. It should stick to the leaf. Can we protect it from rain? Do we have the features that it is very easy to adopt and makes commercial sense to the growers as well? So with that in mind, we developed what is a platform which is called BioClay. Really proud of this platform now, in which what we have is bio. The word bio is the biological active, or the RNA, or the RNA interference part of it. And clay is nothing else, but it is like a fancy postman, a carrier that delivers this double-stranded RNA. And this particular invention it was done along with Professor Gordon Zhu, who is a nanoengineer at the University of Queensland, and Professor Max Lu, who was earlier at the University of Queensland, but now he's Vice Chancellor, University of Surrey in England, and he's also a nanomaterials expert. So to me, this technology was also really important, because to me it signaled the combination or the power of cross-disciplinary science. So it was about nanotechnology and biology or biotechnology coming together to develop at least, or think about a platform that can work as a stable application of RNAi. Once this particular project actually was first funded by Bill and Melinda Gates Foundation. As you can see here, my postdoc is really fond of James Bond, and I cannot shrug off this picture from my slides. The whole idea is, yes, using these clay particles to deliver RNA interference. The bioclay, the trigger molecule, of course, double-stranded RNA, and the clay that I'm talking here about in this particular presentation, we have other clays as well now, is the layered double hydroxide, which is just a positively charged anionic material. The schematic of BioClay is that we load it on these double-stranded RNA on these clay particles, and that's what provides us it with the features of stability and long-lasting feature. Once sprayed as a foliar application on the leaf, this clay actually starts degrading as a very natural mechanism on the leaves. In the presence of moisture, there may be a formation of carbonic acid, which is one way of this releasing this double-stranded RNA from the clay particles. And also this clay also degrades in the presence of moisture as well. So there are two ways that the double-stranded RNA can be released on the leaf surface. And of course, then it has to enter into the leaf system in order to afford protection and trigger off the RNAi pathway. The clay, as I mentioned, is a layered double hydroxide. It's positively charged nanosheets. Average size is about 45 nanometer. Now, this is one of the clays that we really started with and have done quite a quantum of work, but as I mentioned, we do have other clays as well, depending upon the application, whether it is a chewing insect application or whether it is a fungal application suiting to the purpose, and also depending upon the crop host as well and the point of application as well, where you would like to apply this topical RNAi. One of the first step, of course, was that we should be able to load this double-stranded RNA onto the clay particles. And this is just an anionic binding. There is nothing much to it. It's absolutely, the beauty of this technology lies in its simplicity for me. I'm not a nano person, so I was very, very worried when the nano engineer said and walked into my lab and said, look, here is this chalky white solution and you can load your double-stranded RNA onto it. And it's just adding that into this solution, shaking it for 4 hours, and yes, it's bound to the clay particles. And this is one of the images, one of the very rare images we could take using the microscopy, because if with the heat, the RNA actually degrades if it's a very high heat. But this was one of the images that we could capture showing the loading of the double-stranded RNA onto these clay nanosheets. This gel once again shows the loading, and we tried different sizes. This is for the pepper mild mottle virus, which was about 900, around 900 kilobase pairs. This is CMV, cucumber mosaic virus, about 300 base pair construct. And as what you will see here is when the RNA is loaded fully on these clay nanosheets, it does not migrate into the gel. So we tried different ratios of LDH or the clay versus the double-stranded RNA. And as you can see here, maybe at a ratio, this was like 1:1, like 1 LDH, 1 part dsRNA. This is 1:2. At 1:3 and 1:4, we start getting complete loading. That is, we see the fluorescence only in the well, and we cannot see any double-stranded RNA migrating into the gel. These were the pepper mild mottle virus, the 1 kb1 is all in vitro transcribed. We have also tried bacterially expressed very crude extracts. As you can see here in the lane that it's a crude dsRNA, it has multiple bands, but once again, it loads completely on the LDH nanosheets. The next key question before we moved into the glasshouse trials was that, is this BioClay doing the functions that we would like it to do? And therefore, we tested the stability by treating it with nucleases or showing it in the plants in the glasshouse. And you can see that this was stable even after 30 days of the spray as compared to naked dsRNA, which did start to disappear at 10 days. We also did labeled the dsRNA with Cy3-fluorophore, and once again washed the leaves. As you can see here, this is naked before and after wash. We really did the rain simulation experiments, and this is before and after wash when it is sprayed as BioClay. So it does adhere to the leaf surface and is stable. This is one of the slides. We have lots more data which we are soon going to publish, where we are working on the entry of dsRNA into the plant cells. And as you can see here, that it does enter, and this is the fluorescence that we can see into the veins. And we have done some further work using Z-stack and other analysis showing that double-stranded RNA enters after a foliar spray application of BioClay. This is a classical slide. Once again, I reached out to Francis Tanlado, and I'm very grateful to him. He did send me the original double-stranded RNA construct that he used in his experiments with pepper mottle virus. And as you can see here in this panel, the spraying was done on day 0, and the virus was challenged on day 20. So we can see that even after 20 days, this window of protection was there, and we have now extended this window of protection to about 30 or 40 days that we have been able to work on. There is also, we tried to do some experiments on new growth as well. That is, the plant was sprayed and then we marked the leaves which were sprayed, but challenged the virus, in this case cucumber mosaic virus, on the new leaves. As you can see here, if you look at the panel C here, this is water treated plants, this is clay treated plants. And even when challenged on the new leaves, we could see that only 1 plant out of 20 tested or 16 tested was infected. And furthermore, we have done work which really shows the translocation of dsRNA into the new systemic leaves. So there is a movement of dsRNA within the plant system. We are still trying to answer a little bit more on whether apoplastic versus symplastic movement of double-stranded RNA in the plant system, as well as the bidirectional movement based on the application. This sort of sums up in a way that yes, you know, from co-inoculation experiments or spray application and 5-day window protection, we have gone up to, and still this is once again, much more work has been done now in this space, not only by us, but number of large groups. It's really becoming a wealth of information from Guy's lab, from other people working on different things, which have come out in the support of this particular area. This just captures some of the examples. By no means it's really that the work is happening on fungi. Work is a huge body of work is happening in insect pests, especially chewing insect pests. Although, and everyone is also talking about the issues with lepidopterans, and little bit of information is also emerging on sucking insect pests such as whiteflies and aphids. We are also collaborating with Professor Hailing Jin at the University of California, Riverside, and this is one of her slides which shows that double-stranded RNA spray working for botrytis, and this opened up a window for consideration for post-harvest diseases. And once again, she had the issue that it was the very short protection window, and we have done experiments now which shows that BioClay can extend this short protection window even on detached leaf assays on some or some of these assays that Professor Hailing Jin is conducting in her lab. Okay, let's see if it advances. Seems to be a little stuck here. I'm almost at the end of my talk. I hope I'm going okay with time. Bruno, Jeremy, let me know. As I mentioned on fungal diseases, we have recently got a big hub, which is an Australian federal government funding, along with different industry partners. So we've got about $18 million in cash and in kind. We have different partners involved, and we really welcome more collaborations into this hub, not only for fungal diseases, but in any of the crop pests that we're working with. I'm really open to any collaborations where you would like to test our delivery system for your pest or pathogen that you're working with. In Australia, the Office of Gene Technology Regulator has actually passed this legislation that it is non-GM. It took me 2 years. It was number of trips to Canberra here, where the Office of Gene Technology Regulator is located, lots of data presented to them. And finally, it was legislated and passed in the legislation in November late last year that yes, any topical application of RNAi in Australia is regarded as non-GM. And I've been also involved in conversation with OECD and discussing with various OECD partners in this space, not only about the non-GM nature, but other regulatory aspects of RNAi-based biopesticides. So for me, the next big thing is really small, and design of regulation and public opinion are really crucial for the adoption of these technologies. So we must bear this in mind, not only about regulation, but also about social licensing if we aim to make this technology successful. We are trying it as topical spray or tissue culture or seedling application in baits and feeds. I say that the possibilities with this group are endless. So you have a wonderful group here, whether it is viruses or fungi or different insect pests, huge potential in protected cropping, pack houses. We are also exploring, actually extended this to animal health as well. So that has opened up another window of targeting some of the pests and pathogens and the biosecurity issue. Thank you very much from the BioClay team to answer any questions. And thanks once again for this opportunity.
So thank you very much, Nina. It was a very interesting talk and a very good introduction also to the RNAi technology and so on. We have a couple of questions. First of all, how does the double-stranded RNA enter the plant cells from the sprays?
Yep. So this is something which we are looking more deeper into it as well. So there is always a balance of passive versus active transport. With clay, particularly, we are just looking at passive transport. It depends upon the number of stomata on the leaf surface. Most of the time that we have done the confocal imaging in microscopy work, it looks like that is the channel for the entry into the leaf surface or into the within the cells. And then there is a movement. You know, we have seen that it goes to up to about a depth of more than 30 micrometers or more than 50 microns. And we have also seen that when you apply it on the adaxial surface or the upper surface of the leaf, you can then detect it on the lower surface of the leaf when you do the imaging with the stack. But entry is passive through natural opening.
Okay. Another question, they're coming in thick and fast now. Did you check, excuse me, did you check the processing of the double-stranded RNA by sRNA-seq in sprayed plants prior to virus challenge?
Prior to virus challenge, yes. So definitely that was, and again, I think with most of the groups and most of the papers I have read, the double-stranded, the detection of siRNAs or small interfering RNA seems to be an issue. It could be, I have a feeling, and most of our work is showing that there could be a new pathway that we are looking at here instead of when I say new pathway, it is the different kind of processing pathway. Our work shows that double-stranded RNA hangs for a much longer period before it gets processed. There could be, and especially with clay, because it's such a slow release mechanism, what we need to do is capture a bit of a window with the detection limit for siRNA. So we are now using nanopore technology to answer some of those questions and doing some really sort of timed course experiments starting from 5 minutes after the spray and going to about 72 hours after the spray using that nanopore sequencing technology. So I'll be happy to share more in the next 6 months or so.
Okay, there's two related questions here. How does the clay bind to leaf surface? And are there differences between plants because of the different leaves and leaf types?
Yeah, thank you. To be honest, I wouldn't be able to comment how it binds, like it just sticks to the leaf surface, and I wish Gordon was there to answer more about the LDH chemistry. But to the second part of the question, yes, we have tried multiple hosts. So we are working on capsicum, zucchini, peanut, canola, cotton, whichever host the stability. And our very first thing is to test, that whether it sticks to the leaf surface. And yes, it remains there for about 14 to 20 days before we start doing any biology experiments. So far, we have not seen any variation in terms of adherence and leaf surface.
Okay, I think we'll just take one more question. Somebody wanted to know the ratio of double-stranded RNA to the clay nanoparticles. Could you repeat what is the optimum ratio for the mix of the clay and the double-stranded RNA?
Depending upon, it's a little bit variation there, depending sometimes on the purity of the dsRNA as well. So I would say it is very important to keep in mind that the double-stranded RNA purity especially for loading or clay particles, can have an influence. But in most instances, the loading ratio is 1 part dsRNA and 4 parts clay.
Okay, I think we've given this one a lot of time now. So I'd like to thank you again, Nina, very much. I take it you're happy to receive any other follow-up and any other questions as they come through, and we can email those to you, or people can contact you directly. Bruno,
Thank you very much.
Sorry, Bruno, did you want to say a quick word before?
Oh, well, I want to say thank you to Nina for sharing this time with us, and I really hope we can remain in contact and have another opportunity to meet at least on virtual system like this, or in the future maybe also some possible collaboration. Thank you again for joining us.
Okay,
thank you very much.
Thanks very much, Nina. Now we'll move on to the next presenter, which is Kat Capel. Are you ready, Kat?
Yes, I am. Let me just share my screen for